Today I'm actually going to talk about a specific technology development out of my laboratory that I'm working on right now, and I guess you could say I'm kind of in the valley of death as we speak, and I'm not alone, but we still need new partners to come down in there with us and, and help us to determine whether this technology itself is actually going to make it out or whether something related to this technology will end up making it out. So we're still in the development phase, and I'll go through in the slides talking a little bit in detail about sort of how the engineering development phase proceeded. And then at the end, I'll show you the prototype as it currently exists. And you can see that prototype development and design is inherently an iterative process that results in imperfections and some imperfections that you don't notice until you actually make something and hold it in your hand. And so as engineers and as designers, that is what we're focused on. That's the process that we're focused on. And we want to try to get as many voices and as many opinions involved in that process as we can. And so the folks who would eventually hopefully use our devices are very important in informing that process. So let's see if we change. We do. Okay, so normally in my laboratory, what we work on are fully integrated microfluidic devices for point-of-care diagnostics, which is a large way of saying we try to do molecular diagnostics from sample input to answer in one little credit card-sized device. Okay, and most of what we've worked on to this point involves, all of these steps, oops, now I'm in the wrong place. All of these steps of sample collection, filtration of the sample, sometimes if there's cells we have to do cell lysis, isolation, purification of DNA. If we have RNA, we have to do reverse transcription, amplification, and finally detection. This is usually the pathway that we follow, and the projects in our lab range everywhere from influenza A diagnostics, so taking nasopharyngeal washes and putting them in this thing and coming out the other end with an RT-PCR output that tells us whether or not someone is infected with influenza A, all the way through infectious diarrheas, where we start with stool samples and we do the same kinds of extractions on those samples, downstream looking at other amplification technologies, some of which are standard PCR, some of which are isothermal amplifications, and finally doing detection. And all of these things are great, and they're small, and you can hold them in your hand, but the instrument that runs them is about as big as this podium. So it's not really a point-of-care technology at this point, and it requires a lot of engineering input, automation engineering, and it requires a lot of investment and time, money, interest from now the commercial community to make these technologies really go forward. And while these problems continue to be very interesting to us as a laboratory, and we focus on them from a basic science, molecular biology point of view, we wanted to talk about, as a group, well, what can we do now? What have we learned from making these things that allows us to do something that would impact global health sooner? And the part that we are good at are these sample preparation steps. So we decided one day to take basically just the sample preparation steps out of that complicated system, and also out of the laboratory, away from a centrifuge, away from pipetters, away from all of the complicated equipment that you have to put together to do a sample prep, and also away from a cold chain as well. And do sample preparation or extraction of the nucleic acids, RNA or DNA, from a human sample at the point of care without requiring any plug-in power source, okay? So we see, along with, I'm jumping a little bit ahead toward when we got funding for this project, but this project is currently funded by PATH here in Seattle. And through discussions with them upon funding this project, we determined that HIV viral load testing would be a good first demonstration for this technology, because you're using a whole blood sample, you're trying to get RNA, and if you can get RNA and preserve RNA at the point of care, and you can do better than plasma transport, which requires a cold chain, or dried blood spot analysis, which has a rather high limit of detection, then perhaps you can start to affect change on the ground in terms of treatment of a particular patient. And it also has the possibility to make diagnosis possible for infants, for instance, that are not ELISA diagnosable until 18 months. So our goal, again, as I said, was to eliminate or reduce the cold chain. We wanted a low detection limit. On the first prototype, we gave ourselves a 500 copy per mil detection limit. We believe this would have a therapeutic impact early. Of course, we have to go lower than that in the next prototype. There's also a potential, because we're not eluting the nucleic acids into a cold storage needed form at the point of care. There's also a potential for using this technology for long-term banking of nucleic acid samples with minimum infrastructure. So the basic principle, again, is do this at the point of care and store them in a shippable, bankable way, where you could, which is independent of downstream processing. So you don't have to do PCR necessarily. You don't have to do HDA. You don't have to do a branch DNA assay. You could do any one of those things, and the RNA that you elute from these columns, which I'll show you in a second, is elutable in a volume that's appropriate for your downstream assay, or in a buffer if it's a low salt buffer that's appropriate for your assay. And that was important. So the process flow, the chemistry, I won't go into too much detail about the chemistry here, but what's happening for those of you who do this kind of work is it's simply based on boom chemistry. It's like a QIAGEN extraction kit, except you're not using a centrifuge. You're using a bicycle pump, actually. You start with a column, which we call our solid phase extraction column. We then we load the sample onto the column in the presence of a lysis buffer. The lysis buffer aids in the binding of the nucleic acids preferentially to this column. We then come through with a wash buffer. And then at this point, we dry the extracted nucleic acids in place on the column. If we're using RNA, we add a preservative, and that tends to be, that seems to be extremely effective for preserving RNA at high temperatures. And then the sample can be shipped, and once it reaches its location, it can easily be eluted with water from a pipette or a pump like the one we have into downstream assay flow. So I like this picture because this is what it looked like when we were sitting at the table saying, man, these microfluidic chips are a pain. Why? The secret is they leak. They all leak. There are some ways of getting around leaking. There are some ways of getting around reagent storage, but it's kind of a hard problem, and it's one of the problems that I think if we threw a whole lot of money at it commercially, we could solve it, just like we solve, it always blows me away that I can have this iPhone that does all these things and it doesn't break unless I drop it in the toilet, but we can't, seal a microfluidic chip reliably for, a year-long shelf life, and we can't do that in huge quantities. So, I said, well, what if we just completely take, can you still hear me? Okay. What if we just completely take out the microfluidic, okay? Because even though that's what, I build my lab on here, let's just get rid of that. And let's make the solid phase extraction column that we make, the plastic column that actually grabs and releases the nucleic acids on a little tube, like a straw. And I'll pass these around, and I made enough, so if somebody steals them, it's okay. They're clean. But what's in this is it's a clear straw, it's hollow, and inside of it is basically a porous polymer monolith that's cross-linked in situ inside of the straw. So we can make these in large quantities in the lab. And they work again like a silica solid phase extraction column, basically. And I said, what if we can eliminate the leaking, and then we can put the reagents up in here, and all we need is a lysis buffer, a wash buffer, and then we'll elute it. And in the original design, I said there'll be a tube down here. We'll elute it into that tube, and then we'll close the tube, and we'll put it and ship it off. And it's just RNA now, or it's just DNA now, so it's not somebody's blood. It's not infectious. Wouldn't that be great? And here's these straws, and they'll be disposable, and they'll be durable. And then that's kind of where I stopped, because my training with AutoCAD is, 15 years old, and I'm not an industrial designer. And I ran into a colleague of mine, Jose Gomez Marquez at IIH at MIT, and he said, I have some industrial designers, and they'll make you a drawing. And this is what it looked like from that picture, and this is fabulous. This still doesn't work. It's just a picture. But it's a great picture. It's way prettier than anything I can make. And this is just one of like 5 different pictures that they made. So it was very exciting to like see, cool, we could do this. There could be a reagent package in here. It's a blister pack. It's got all the reagents. All you do is click through and turn the thing and push the button, and the reagent goes through, and when you're done, there's your RNA. Put it into the silica packet containing, mail, and you ship it off to the lab, right? It weighs nothing, it's fabulous. Okay. And then furthermore, we have a 3D printer, so we made one up. New York Times came and took a picture of that. Still doesn't work. This is the fun, now I've skipped through like 12 slides that I'm not going to show you. But this is what the prototype looks like today, which is what this thing is that's sitting up here. And I don't know what the best place to show this is for everyone to see, but I'm going to do this because this looks like a demo place for science class. So what we end up with is this prototype. So we wanted it to be like a nice plastic container that looked like a bottle, and it had the clicking, and you could turn everything, and it only went one way, and it was idiot proof. But it turns out, in order to prototype this and actually make sure that it works, those things are pretty high end on the design the design continuum. So what we can do is we can go to the machine shop, and we can make devices that don't look as good, but they do all of the parts. So we have been able to, I'll show you the rest of the slides before I show you the demo. This looks pretty bulky, and I told you some of it was disposable, and also any part of this that touches somebody's blood gets thrown away. So what the inside of that routing block looks like, this piece of black sand here with all these screws coming in and out of it, is actually just some open channels. And those open channels are what do the work. So, and these open channels, since we had to build this thing from off-the-shelf parts and valves, these, I don't like this name, reminds me of the Gulf. When I saw this, I was like, we got to come up with some other name. But the internals of this are really quite simple and quite small, actually. If you come up and look at the prototype later, you can see that we could essentially compression mold or injection mold all of these parts together. We could put the monolith or the solid phase extraction column in here using in situ photopolymerization, and then this whole thing gets thrown away after each patient. So, and it would look like a little, it hopefully would be about this big with maybe a couple things coming off of it.